Very warm welcome to our biology class. Students, in this class we are going to understand regarding the spermatogenesis. In our earlier class we have already understood all these things in a very proper way. We have understood internal all the detailed structure of a testis, and we have seen how the seminiferous tubules are located inside the testis, and in between the seminiferous tubules there is an interstitial tissue, and in that interstitial tissue we used to find the presence of immunocompetent cells as well as Leydig cells. And in my previous video I have even explained you how the hormones are going to act on the Leydig cells and testosterone secretes, and what is the role of testosterone in male body. Already I have discussed in my previous video. You can please observe that, please watch that video even to understand the secondary sexual characteristics which are developed with the help of testosterone. So today we are going to mainly focusing on the spermatogenesis. Students, to understand the spermatogenesis in a very proper way, what we will do right now, we will magnify this particular area by considering one nourishing cells called as an Sertoli cell, and even we will take one or two germinal epithelial cells, and we will see that how their divisions are going to occur during the spermatogenesis. For that reason I have magnified this particular area only in the seminiferous tubule. Suppose it is the basement membrane of seminiferous tubule, on that Sertoli cell is located, and I told you we are considering only one or two germinal epithelial cells. Now you need to understand very properly, students, in case of male, whenever the boys will reach to the age of maturity, by that time only hypothalamus, as it is the part of the brain, it comes to know about the physical development of boy, and once it comes to know that that particular boy body has physically completely developed, then and then only hypothalamus will start to secrete GnRH hormone, and GnRH hormone will start to act on the anterior pituitary gland. In response to that, anterior pituitary gland secretes LH and FSH. What is the role of LH? LH will act on the Leydig cells, and Leydig cells will start called testosterone, and this testosterone is the one which is going to develop all the secondary sexual characteristics. What are all the secondary sexual characteristics and how they are going to develop, all that I have already discussed in my previous video. I have given the link in my description box. You can please check that video and understand what is the role of testosterone in male body. And the testosterone is the one which is going to even activate the process of spermatogenesis. We will see now how it is going to activate. By the same time, even in case of male, FSH is secreting, and this FSH is going through the blood and acting on the Sertoli cells. And once the Sertoli cells are got activated by the FSH, then they start to secrete several factors. These factors are plays very important role in the process of spermatogenesis. And there is a one protein factor is going to be produced, that is called as androgen binding protein. It is the another one of the factor which is getting secreted by the Sertoli cells once they are got activated by the FSH. What is the role of this protein then? This protein is the one which will help in making accumulation of the testosterone inside the seminiferous tubule. You need to understand, students, testosterone is the one which is getting secreted by the Leydig cell. It is outside the seminiferous tubule, but where the spermatogenesis occurs, it is inside the seminiferous tubules. Because of that, it is very essential to accumulate this testosterone inside the seminiferous tubules. And how it is going to get accumulated? It is with the help of ABP, androgen binding proteins, which are getting secreted by the Sertoli cells. Now once the testosterone is got accumulated inside the seminiferous tubule, and even there are the certain other factors are got secreted by the Sertoli cell. All together, they are going to activate the process of spermatogenesis. Students, in the spermatogenesis, there is the first phase called as a multiplication phase, where the germinal epithelial cells will divide mitotically and forms the two newly developed cells here. And whatever these newly developed two cells are there, these are called as an oogonia, students. And for your understanding, I am saying, so out of these two oogonia, one oogonia is considered as an A oogonia and other one is considered as an B oogonia. Whatever the A oogonia is there, that is going to continue the further divisions. The B oogonia is again going to act as an stem cells, means like the germinal epithelial cells. Again it is going to divide by the mitosis. So here you need to understand, out of these two, one is going to continue the further division and one is going to act as an stem cells again to maintain the number of cells here. Otherwise what will happen, students? Suppose for example all these germinal epithelial cells at once, if they keep on dividing and leading into the formation of the sperms, then there must be some other cells to be produced, those germinal epithelial cells, to get produced the new sperms or not. So to maintain the cell line here, this is what going to happen. Out of these two, whatever newly formed cells are there, one is going to continue its further division and one is going to act as again the stem cells. I hope you understood how it is going to happen. They are going to keep on dividing by the mitosis and finally leads into the formation of spermatogonia. And whatever this particular first phase is there, students, it is all the mitotic divisions. And as you know that mitotic division, it is the equational division where the number of chromosomes are going to be maintained as it is. Means the diploid cells will lead into the formation of diploid cells only. Germinal epithelial cells are diploid and the spermatogonia is also diploid. And this phase is the multiplication phase, and it is the first phase in the spermatogenesis that what you need to be understand. After the multiplication phase, whatever these all the oogonias are formed now, out of them, some of them are going to grow in their size, because more amount of the cytoplasm started to get accumulated in them, and that of the accumulation is leads into the increase in the size of those cells. And those who are already increased in their size, those spermatogonias are now, we call them as primary spermatocytes. You must remember when to call spermatogonia and when to call primary spermatocytes. This sequence you should have to be properly remember, students. Now, whatever these primary spermatocytes are there, here only the cytoplasmic content has increased, but not the chromosomal number. Yeah? Hence, the chromosomal number remains as it is. And whatever this phase is there, this phase we call it as growth phase. Students, in one of my previous video, even I have properly explained you what this growth phase, actually what going to happen during the multiplication phase, growth phase, that all the details I have discussed in one more my previous video. Now, these whatever the primary spermatocytes are there with a diploid in chromosomes, these are going to divide by meiosis. And as you know that the meiosis is divided into the meiosis 1 and meiosis 2. Now the meiosis 1 is going to occur in these primary spermatocytes, and that leads into the formation of the two equal cells. Means even in the number of chromosomes also, and as well as cytoplasmic content also. This leads into the two equal cells, and whatever these newly formed two cells are there, these are called as secondary spermatocytes. But the secondary spermatocytes are produced by the meiosis 1, and meiosis 1 is the reductional division. Hence, the number of the chromosomes are going to reduce here. Only 23 chromosomes in this and 23 chromosomes in the other cell. This is what going to happen, students. And whatever these secondary spermatocytes are there, once again they will divide, or both these cells leads into the formation of 4 cells here. And all these 4 cells are formed by the meiosis II, and meiosis II is the equational division. Hence, whatever the number of chromosomes we find in the secondary spermatocytes, all they are going to be observed in the spermatids, and newly formed cells are called as an spermatid, students. And these are also haploid in the chromosomes. And whatever these all newly formed spermatids are there, all these spermatids are leads into the formation of spermatozoa. Yeah, they are leads into the formation of finally spermatozoas are forming. But are you able to understand here, students? These are normal just cells with a nucleus, cytoplasm, plasma membrane, and all other cell organelles inside it. But they are not at all have a safe of sperms. And they need to go through the process. And after the process they gone through, then and then only they are going to become like a sperms. They are going to get the tail, midpiece, head region, acrosome, all that things are going to form in the sperms. So whatever that process of transforming the spermatids into the spermatozoa is there, that process is called as an spermiogenesis. Students, you must have to understand spermatogenesis is different, spermatogenesis is different, and spermiogenesis is different. Spermiogenesis means transformation of spermatids into the spermatozoa. What is the meaning of it, students? You might be observing these are the just normal cells, like they don't have a tail like structure, they don't have a head like structure, they don't have a mid-piece like structure, but all that slowly going to occur, and all these things are going to occur with the support of Sertoli cells only, and finally they lead into the formation of spermatozoa. So here whatever this primary spermatocytes are there, to become a spermatozoa is there, this is called as an maturation phase. And here the diploid spermatozoas are formed, students, and this phase is called as an maturation phase. You must remember the sequence of phases also even. First phase in the spermatogenesis is multiplication phase. Second phase is growth phase. Third phase is maturation phase. And after the maturation phase, you might be observing here we used to find the formation of spermatozoas. And students, I told you spermatozoas are not freely present here. They are found attached with the Sertoli cell, because it is the one which is acting as a nourishing cells. It is providing all nutrition to the dividing all these cells by providing the factors, nutrition, everything which is required for all these cells. And finally, whatever these spermatozoas are formed, these spermatozoas are found attached to the Sertoli cells. They need to be released from the Sertoli cell. And you might be observing now they are got released. The process of release of a spermatozoa from the Sertoli cell is called as spermiation. You must have to understand the meaning of all these terminologies, students. Spermatogenesis is the process of formation of sperm. Spermiogenesis is the process of transformation of the spermatids into the spermatozoa. Spermiation, it is nothing but release of spermatozoa from the Sertoli cell into the lumen of seminiferous tubule. This is what going to happen, students. Now we will understand how all this process of spermatogenesis is controlled by the hormones. Here the hypothalamus, here it is the seminiferous tubule. Inside that Sertoli cell is there, and number of germinal epithelial cells are there. And outside to the seminiferous tubule, we used to find the presence of Leydig cells. Suppose now, whenever the boy has reached to the age of maturity, again, let me remind you, students, once again, boys, whatever that maturation is there, that maturation may not be occurs exactly on the 12th year. It may be during the 13th year or 14th year of his age. Why it is happens like that? Why not exactly 12 years in all the boys? Because I told you even in a earlier, just in the previous slide. So hypothalamus is the one which will recognize whether his body has become fit to go for the reproduction or not. If the hypothalamus, part of the brain, found that his body has become fit for the reproduction, then and then only the brain will make him to become and reproductively fit. Now let us understand how the hormonal regulation occurs here. Hypothalamus, at the age of puberty, it will start to secrete the GnRH hormone, and this GnRH hormone will act on the anterior pituitary gland. In response to that, the anterior pituitary gland secretes FSH and LH, and this whatever the LH is there, it is started to act on Leydig cells. And once the Leydig cells are acted by the LH hormone, then they started to secrete the testosterone. And this testosterone is the one which is responsible for all the secondary sexual characteristics. And simultaneously, whatever FSH is there, it is going to act on the Sertoli cells. And the Sertoli cells are secreting many other factors. Even the Sertoli cell is secreting androgen binding protein. And this androgen binding protein will help in making the concentration of testosterone inside the seminiferous tubules. And once the testosterone is got into the seminiferous tubule, it will activate the process of spermatogenesis. This germinal epithelial cell will keep on dividing, and finally it will lead into the formation of spermatozoa. But remember students, whatever this complete entire process of division is there, spermatogenesis is there, it is under the control of hormones, FSH also and LH also, means ultimately the testosterone also. Suppose if the testosterone concentration increased very high, then what will happen? The process of division is going to occur very fast. And if the process of divisions occurs very fast, then whatever the forming spermatozoas are there, they may not be normal. They may lead into the abnormal sperms. Because of that, concentration of the testosterone also should be maintained properly. How the concentration of testosterone maintained? It will maintain by its own. Suppose whenever the testosterone concentration increase in the blood, automatically that increased concentration of the testosterone will start to give the negative feedback to the hypothalamus, and even it will give the negative feedback to the anterior pituitary gland. Because of that, LH hormone secretion is going to reduce, and once the LH hormone secretion reduce, automatically testosterone concentration is going to reduce. In the same way, whatever this FSH concentration is there, it is maintained with the help of secretion of the one more hormone called as an inhibin. This inhibin is secreted by the Sertoli cell. And when the inhibin is going to secrete it, whenever the FSH concentration is become more than normal, then and then only it secretes the inhibin hormone, and this inhibin hormone will give the negative feedback to the anterior pituitary gland to reduce the secretion of FSH. This is what the way how the control of testosterone and the FSH is going to occur in the male body. And remember students, once the spermatogenesis is begun during the age of puberty, it is going to continue throughout the life. During the old age also, we used to find the normal spermatogenesis. There is no variation. Suppose if that particular old age man, if he is not suffering from any other diseases, if he is physically fit, then even in him also spermatogenesis is completely normal. There is no any issues with that. Then we need to understand students, whatever this entire process of spermatogenesis is there, it is going to occur in 72 hours. Got it? So complete process of the spermatogenesis will occur in 72 hours. I hope so students. By this class, you are properly understood how the spermatogenesis occurs and how hormonal regulations are going to take place. Suppose for example, in the theory exam, if they ask you to sketch and represent the spermatogenesis, you have to sketch and represent the spermatogenesis like this. And if they ask you the questions on the hormonal regulation, you must have to remember this. And you should have to remember all these cells in the proper sequence. germinal epithelial cells, spermatogonia, primary spermatocytes, secondary spermatocytes, spermatids, spermatozoa, and then the final sperms. And remember students, in the seminiferous tubules, sperms are just only going to produce, but their maturation occurs in the epididymis. That things we will understand in the next classes. And even you must have to remember in what chromosome numbers are there in every cells. Yeah, you need to understand that what mean number of chromosomes are there in the germinal epithelial cells and spermatogonia, primary spermatocytes, secondary spermatocytes, spermatids, and the spermatozoa. And sometimes they may ask you the question, suppose for example, if you take 100 primary spermatocytes, then what many number of sperms are going to produce? So easy, very easy answer. 100 primary spermatocytes we have taken. Then if the meiosis 1 occurs, then 200s are going to be formed. And if the meiosis 2 occurs, 200s are going to become a 400. So if we consider 100 primary spermatocytes, then we are going to get 400 spermatozoa. Sometimes they may ask you the question, suppose if we consider 100 secondary spermatocytes, then what mean number of sperms or spermatozoas are going to be produced? If the 100, only one division is left over here. So finally, we are going to get only 200 sperms. That is what the way how even questions are going to come on this particular part. I hope so students, this entire discussion has become very useful for you to understand the process of spermatogenesis. Please the like, and even if you are willing that this lecture has to be reached to the many other students, please share it to your friends also. And if you're not yet even subscribed, please do subscribe, and even ask your friends to subscribe also and support the channel. Thank you once again.